Good morning, everyone. Today, we will analyze and discuss questions from past year exam paper. We will go through each question in detail to understand the concept and ensure we are well prepared for any similar questions that might appear in future exams. Without further ado, let's get started. Which definitions of the magnifications of a drawing of a leaf is correct? The correct definitions of the magnification of a drawing of a leaf is the size of the drawing of specimen in comparisons to the actual size. The electron micrograph shows part of two cells. Which label features identify these cells as eukaryotic? The answer is W only. Plant cells are fixed, stained, and viewed using a student microscope. The light source was natural light. What would be clearly visible at multiply 400 magnification? At multiply 400 magnification using a student microscope, nuclei would be clearly visible. At this magnification, you can typically see larger cell structures like the nucleus within the nucleus, but structures such as mitochondria, grana of chloroplasts, and ribosomes are generally too small to be clearly resolved at this level of magnification. Which lengths are equivalent to 1 micrometer? The length equivalent to 1 micrometer is number 3 and number 6. The reasoning, 1 micrometer is equivalent to 0.001 millimeters, and 1 micrometer is also equivalent to 1000 nanometers. Some secretory cells synthesis and release glycoproteins. What is the correct order of the sequence of events as they occurred in the secretory cells? The correct order of the sequence of events in a secretory cell that synthesizes and releases glycoprotein is 3, 4, 2, and 1. 3 is referred to the statement mRNA binds to ribosome. This is where the process of translating genetic information into proteins begins. Statement 4, synthesis of glycoprotein in the rough endoplasmic reticulum and processed in the Golgi apparatus. Statement 2 is referred to the product accumulates in secretory vesicle. The glycoprotein is packed into secretory vesicle. And statement 1, exosynthesis, is the secretory vesicle fused with the plasma membrane, releasing the glycoprotein outside the cells. Scientists carry out an experiment to separate the organelle in an animal cells by mass. The scientists mixed the cells with a buffer solution which had the same water potential as the cells. He then broke the cells open with a blender to release the organelle. The extracted mixture was filtered and then spun in a centrifuge at a speed to separate the heaviest organelle. This sinks to the bottom, form a solid pellet 1. The liquid above pellet 1 was poured into a clean centrifuge tube and spun in the centrifuge at the highest speed to separate the next heaviest organelle. This organelle sinks to the bottom to form a solid pellet 2. He repeated this procedure twice, more to obtain pellet 3 and pellet 4, each containing a single organelle. What is the function of the organelle extracted in palette 3? The answer is digestions of whole organelle. What is the general formula for amylopectin? The general formula for amylopectin is C6H10O5. Amylopectin is a branch polysaccharide made up of glucose unit, and its general formula reflects the repeating unit of a glucose polymer minus water molecules lost during polymerization. Which statement describes how the molecular structure of starch is suited to its function? The statement that best describes how the molecular structure of starch is suited to its functions is the amylose-amylopectin complex is insoluble and does not affect the water potentials of the cells. This is because starch, which consists of amylo and amylopectin, is an insoluble polysaccharide. Its insolubility helps to prevent it from affecting the water potential of cells, which is important for maintaining cell stability and function. Number 9, cow and whales are mammals that produce milk to feed their babies. Newborn whales grow faster than newborn cows. The milk of both cows and whales contain saturated fatty acids with different chain length. The table shows the percentage of saturated fatty acids of different length in cow and whale milk. Which statement about the ratio of short fatty acid for until 16 carbons to long fatty acids, 18 or more carbon in the milk of cows and whale is correct? The ratio in whale meats is lower because young whales need more energy than young cows. The structure of phospholipids and triglycerides include the following. Statement 1, glycerol linked to fatty acid. Statement 2, hydrophobic fatty acid change. Statement 3, saturated fatty acid change. Which structures enable the formations of a lipid bilayer in cell surface membrane? The structure that enables the formation of a lipid bilayer in cell surface membrane is statement 1 and 2. Phospholipids have a glycerol backbone linked to fatty acids with hydrophobic fatty acid change. These hydrophobic fatty acid change are crucial because they form the interior of the lipid bilayer, while the hydrophilic glycerol head face the aqueous environment on either sides of the membrane. The diagram shows the molecular structures of a peptide. The answer is C. The diagram shows a hemoglobin molecule. Which row identify the different parts of the molecule? The answer is B. 1 is referring to binding site. 2 is hydrophilic amino acids. 3 is alpha helix. And number 4 is hydrophobic amino acids. Bacterial cells divide by a process called binary fission. Which macromolecule must be synthesized for binary fission? For bacterial cells to divide by binary fission, the macromolecules that must be synthesized are statement 1 and statement 2. The reason is below. Reason number 1, cell membrane protein and RNA. These are necessary for various cellular functions and replication processes. Statement 2, DNA and peptidoglycan. DNA is crucial for replicating the genetic material, and peptidoglycan is needed to synthesize new cells wall for the daughter cells. And for statement 3, enzyme and cellulose Enzymes which are protein are also involved, but enzyme and cellulose are not specific to bacterial cells division. Cellulose is a plant polysaccharide and is not relevant for bacterial cell wall. A student carried out experiment to investigate the effect of enzyme concentration on the rate of hydrolysis breakdown of protein in milk. When the enzyme and milk were mixed, the protein was hydrolyzed and the mixture changed from cloudy to clear. The student investigated 5 different enzyme concentrations and recorded the time taken to reach the endpoint for each. What is an appropriate control for this investigation? The appropriate control for this investigation is carrying out a further experiment where the enzyme solution is replaced with water. What determines the specificity of an enzyme? The specificity of an enzyme is primarily determined by the bonding between the R groups of the polypeptide chain. And this bonding helps to form the enzyme active site, which is crucial for binding to a specific substrate. Therefore, the correct answer is C. The graph shows the effect of temperature on the rate at which the enzyme in a biological washing powder digests and removes fruit juice stain. Which statement explains the shape of the graph at temperatures higher than X? The correct answer is statement 1. Bonds are broken between the R group of amino acid in the polypeptide chain of the enzyme. Statement 3. The tertiary structure of the enzyme is altered. And statement 4. The shapes of the active site and the substrate are no longer complementary. What describes a carrier protein in cell surface membrane? The description that best fits a carrier protein in cell surface membrane is a glycoprotein that is involved in moving substances through the membranes by both active and passive transport. Carrier proteins are integral membrane proteins that bind specific substances and transport them across the membrane, utilizing either active transport, which require energy, or passive transport, which does not require energy. What would happen to a typical bacterium when it is placed in surroundings which have a less negative water potential than that inside the cells? When a typical bacterium is placed in an environment with less negative water potential, example, a hypotonic environment, compared to the inside of the cells, water will move into the cells due to osmosis. The cell wall of the bacterium provides structural support and help prevent the cells from bursting, although some pressure may be exerted. Therefore, the most accurate description is there will be a net movement of water into the bacterium. By which process do hydrogen carbonate ions leave red blood cells? Hydrogen carbonate ions leave red blood cells primarily through the process of facilitated diffusion. Specifically, this involves the exchange of hydrogen carbonate ion with chloride ions via an antiporter protein known as the chloride bicarbonate exchanger or BAN3 protein. So the correct answer is facilitated diffusion. In an experiment, fluorescence dye were used to label protein on the outer surface of cell surface membrane. Living human cells were labeled with one color of fluorescent dye and mouse cells with a different color of fluorescent dye. A human cells and a mouse cells are then fused to form a hybrid cell. At first, the different fluorescent labels remain separate, but after 40 minutes, they are distributed randomly across the hybrid cell surface membrane. What does this experiment show? This experiment shows protein move freely in the phospholipids of a bilayer. The diagram shows the mitotic cell cycles, during which phase is DNA replicate. This is a similar question which is already appeared in October 2017. But the questions is related to chromosome, condense and become visible. But for this May 2017, the question is asking is related to during which phase is DNA replicated. In the mitotic cell cycles, DNA replication occurs during the S phase, which is part of interphase. A likely represent late anaphase or telophase, not relevant for DNA replication. B likely represent G1 phase, just before DNA replication start. C is likely metaphase after DNA replications has occurred. And D is typically S phase where DNA replication occurs. The electron micrograph show a group of human chromosome. Which label is correct for each of the structure label W, X, and Y? Y is represent chromatic, X is represent telomere, and W is represent centromeres. Which statement about the behavior of chromosome during mitosis is correct? The correct statement about the behavior of chromosomes during mitosis is they condense to prevent further translation of gene. During mitosis, chromosome condense to become more compact and visible under microscope. This condensation helps prevent the chromosome from becoming entangled and ensure that they are accurately distributed to the daughter cells. What is the smallest unit of DNA molecule that can be altered by a mutation and cause a change to the coding of polypeptide? The smallest unit of DNA molecule that can be altered by a mutation and cause a change to the coding of polypeptide is nucleotide. A change in a single nucleotide, such as a point mutation, can affect the codon sequence and potentially alter the coding of a polypeptide. Which statement about tRNA are correct? The correct statements about tRNA are statement 1, 2, and 3. Statement 1 is contain base pairing. Statement 2 contain hydrogen bond. And statement 3 is single-stranded. tRNA, transfer RNA, contains base pairing and hydrogen bond, which help to maintain its structure, and it is indeed single-stranded. The table shows the role of 4 different proteins involved in DNA replication. We show the functions of this protein. The answer is D. Which type of sugar and which type of bond are found in DNA molecule? In DNA molecule, the type of sugar and type of bonds are: type of sugar refer to non-reducing, which is specifically deoxyribose. And type of bond is referring to hydrogen between complementary base pair and phosphodiester between sugar and phosphate group, though this was not listed as an option. Given the option, the most accurate statement is type of sugar is a non-reducing and type of bond referring to hydrogen. Number 28 is similar questions which appear in March 2019 and October 2017. Refer to this diagram. This diagram represents transverse sections of 3 plant organs. And 2 for the root, Number 2 is referred to folium and 1 referring to xylem. But for stem, number 4 refer to xylem and number 3 is a folium. But for leaf, 5 is referring to xylem and 6 is referring to folium. Which row is correct for folium? So the answer would be C. Root is 2, refer to folium, and for the stem is referred to 3 is a folium, and for the leaf, number 6 is referred to the folium. Sucrose moves from a folium sieve tube elements into a root cells, which changes to the water potential and the volume of liquid in the folium sieve tube elements are correct. When sucrose moves from the folium sieve tube into a root cells, water potentials decreases, become lower in the sieve tube because sucrose increases the osmotic pressure. And volume of liquid decreases in the sieve tube because water moves out of the tube into the root cells. Thus, the correct answer is C. Which statement about water movement in plants are correct? The correct statements about water movement in plants are: water can pass through cellulose cells wall. This statement is correct. Yes, cellulose cell walls are permeable to water. Let's look into statement number 2. Water can pass through lignified cells wall. Lignified cells wall are less permeable to water compared to cellulose cell wall, but water can still pass through them, often through specialized structure like pits. Statement 3, water cannot pass through suberin in cell wall. Suberin is a hydrophobic substance found in the cell wall of certain tissue, example cork, that prevent water passage. So the correct answer is statement 1 and statement 3. The photomicrograph is a transverse sections of a leaf. Which features are characteristic of xerophytes? The answer will be number 1, number 2, and number 5. A maize crop is successfully growing in a field in which the water potential of the soil is minus 40 kPa. What is the most likely water potential of the cell sap in the root hair cells? For a maize crop growing in soil with a water potential of minus 40 kPa, the water potential of the cell sap in the root hair cells is most likely more negative than the soil water potential to facilitate water uptake. Thus, the water potential of the cell sap would be A. The concentration of the heart is coordinated through electrical impulses passing through the cardiac muscles. Which is the correct order of part of the sequence of these impulses? The correct order of the sequence of electrical impulses that coordinated the concentration of the heart is sinoatrial node, 2 right and left atria, 2 atrioventricular node, and 2 Purkinje tissue. This sequence ensures that the electrical impulses first stimulate the atria to contract, then travel to atrioventricular node, and finally propagate through the Purkinje fiber in the ventricular walls to coordinate ventricular contraction. Which features enable the aorta to withstand ventricular systole? The features that enable the aorta to withstand the high pressure during ventricular systole are collagen fibers and elastin fiber. Collagen provides strength and structural support, while elastin allows the aorta to stretch and recoil, accommodating the surge of blood flow and helping to maintain blood pressure. Which row is correct for pulmonary artery? The correct row for pulmonary artery is blood carried, deoxygenated, muscle in wall thin, and lumen size will be large. The pulmonary artery carries deoxygenated blood from the heart to the lung. It was relatively thin compared to artery like the aorta, and the lumen is large to accommodate the blood flow to the lungs. The graph shows oxygen deassociation curve of adult oxyhemoglobin in different carbon dioxide concentration 1 and 2, which condition could change the shape of curve 1 to the shape of curve 2. To change the shape of curve 1 to curve 2 on the oxygen deassociation curve, you need to lower hemoglobin affinity for oxygen, meaning that shift the curve to the right. And this happens when high carbon dioxide concentration causing a decrease in pH. And this makes hemoglobin release oxygen more easily, shifting the curve to the right. Which characteristic of the human gases exchange surface and the lung maintain the necessary concentration gradient for carbon dioxide and oxygen? The characteristic of the human gases exchange surface and the lungs that maintain the necessary concentration gradients for carbon dioxide and oxygen is good ventilations of the lungs. Good ventilation ensures that the air in the lungs is refreshed regularly, which helps maintain the concentration gradient of oxygen and carbon dioxide, which allow efficient gas exchange. Which identify the tissue presence in trachea. This is also a similar question which already appeared in October 2021. And the questions is related to structure of mammalia gas exchange system. And the answer for these questions will be B. The global mortality figures for some diseases in 2002 are shown in the table. How many millions of people died in 2002 from the bacterial diseases listed in the table? The answer is B. B lymphocytes and T lymphocytes are often unable to respond to the antigen on pathogen that are intracellular parasite. What is the reason for this? B lymphocyte usually can find the antigen or pathogen that are inside cells because they are looking on the outside. T lymphocyte can recognize this antigen, but only if the infected cell show the antigen on the surface. If the antigens are hidden inside the cells and not shown, the lymphocyte can't detect them. That's conclude today's session, and I look forward to seeing you at the next session. Please subscribe to Jom Stabila YouTube channel. Don't forget to turn on notification so you don't miss any updates. Best of luck in your studies and upcoming exam, and wishing you every success. Thank you very much for your support and cooperations.